Hello everyone. Today I will present one of my research, deciphering the novel role of AtMIN7 in cuticle formation and defense against bacterial pathogen infection. Further investigation of the mechanism of AtMIN7 may enable strategies to enhance plant health and yield through the genetic manipulation of plant cuticle, which will reduce the agricultural chemical application to benefit the environment and human health. This is my poster. Firstly, I'll give a brief introduction. Plant cuticle covers most of the plant tissues, protects the plants against different stresses, including the attacks by pathogen infection. Plant cuticle are composed of the cutin and wax. With the development of genetic, molecular, and biochemical approaches, we have a better understanding of the components and mechanism involved in the cuticle biosynthesis. Cutin and wax biosynthesis both starts from the carbon-16 and carbon-18 fatty acids in the plastid. Plastid-generated carbon-16 and carbon-18 fatty acids forms the carbon-16 and carbon-18-CoA in the ER. Carbon-16 and carbon-18-CoA will converted to the cutin and wax precursors, and these precursors will be modified and exported from the ER, move across the plasma membrane and the cell wall, and finally deposited into the cuticle layer. So what is AtMIN7? AtMIN7 is an immunity-associated Arabidopsis protein targeted by the HOPM1, a conserved virus protein. In addition, AtMIN7 encodes one of the 8 members of Arabidopsis ADARF-GF family, which are key components of the vascular trafficking system. In our project, we aim to investigate the novel role of AtMIN7 in plant cuticle, and we aim to discover the role of the plant hormones such as SAJ and ABA in cuticle formation in AtMIN7 mutant. We aim to decipher the role of vascular trafficking in plant surface-mediated immunity. We applied different kinds of approaches to study our project, including the genetic screening, hormone treatment, and hormone level determination, bacterial infection, and we apply specific approaches to study plant cuticle, such as staining, metabolic profiling by GCMS, cell imaging through TM and SEM. At first, we got the AtMIN7 mutant and complementation line. The mutant didn't show significant differences of the phenotype, comparing with the wild type and complementation line. Toluene blue can penetrate plant leaves with a defective cuticle. The mutant was stained by the toluene blue, not the wild type and the complementation line. Consistent with the toluene blue staining results, chlorophyll leached faster in the mutant, comparing with the wild type and complementation line. Through TM, the cutin layer of the mutant is much thinner, comparing with the wild type. The cuticle layer of the stomata of the mutant is almost missing, and the stomata structure of the mutant is defective comparing to the wild type, and the percentage of the defective stomata is much higher in the mutant than the wild type. So a mutation in AtMIN7 results in the defective cutin layer and the stomata layer. In addition to the direct observation of the cuticle layer, the leaf cuticle wax and the cutin monomer content were measured by GCMS. The cuticle wax didn't show significant differences, but the mutant showed significant decrease in the total cutin monomer and several main cutin monomer species. So the abundance of the cutin monomers is reduced in the AtMIN7 mutant plants. Plant hormones regulate plant growth and development process. Previous studies highlighted the possibility that plant hormone influence the cuticle formation. Here, mutant plant and wild-type plants were sprayed with different hormones. Only SA and JABA could restore the resistance to TB staining in AtMIN7 mutant, indicating SAJ and ABA could rescue the cuticle formation. We further investigated if the enhanced cuticle formation by SAJ and ABA could increase the resistance of the AtMIN7 mutant plant following the surface inoculation with bacterial pathogen DC3000. The result is consistent with the hypothesis that the restoration of the cuticle by these plant hormones could restore leaf surface defense against the bacterial pathogen infection. We further quantified the hormone levels of the plant leaves and found the leaves of the AtMIN7 mutant contained significantly lower levels of CJ and ABA. In summary, AtMIN7 mutant is defective in leaf cuticle formation, including a thinner cuticular layer, defective stomata structure, and impaired cuticle ledge of stomata compared to those of the wild-type plants. AtMIN7, an important vesicle trafficking component, is critical for cuticle formation and related leaf surface defense against bacterial pathogen infection. Plant hormones can influence the plant cuticle formation. AtMIN7 has the potential capacity to affect hormone biosynthesis and integrate the response to hormones with modification of the cuticle for defense against the bacterial pathogen infection. This is my advisor, Dr. Ye Xia, and these are our lab members. Thanks for their help in my project. We really appreciate the funding support, and our biodiversity starts from ABRC and the Molecular and Cellular Image Center at OSU. Thanks for your attention. Any question is welcome.